The focus of my research has to do with the growth of the kidney during the embryo. And we happened upon a molecule that had a clinical relevance. Its biology is of great interest as well, and turns out to have a lot of completely unknown wrinkles. The biology of the molecule also enhances our belief that it's a very good biomarker of kidney disease as well. So I'll go into all those aspects. Everything I'm going to tell you has been home brewed. We do all of our work, create all of our assays, think really hard about what the next assay is. And so I'm going to tell you about the kidney and why we need these biomarkers and what exactly do we need to do to create a new diagnostic. The first slide starts with 2 clinical cases which you can read. And these clinical cases are certainly not complicated and don't require much medical information. However, they revolve on the fact that the marker creatinine has aspects of nonspecificity. So in the first case, we have someone with a traumatic injury and muscle damage. And a creatinine is 1 which is, quote unquote, the normal range. Medical intervention is required, and a CAT scan is necessary to visualize internal bleeding. And secondly, to examine different tissues that are damaged. So eventually, a contrast with a CT with contrast is necessary. The problem here is that some of these medications and interventions are nephrotoxic, and with a creatinine of 1, you wouldn't get too worried about that, except for the physician who's on his or her toes and has noticed that there's severe muscle damage, which causes rhabdomyolysis, which potentially causes what's called acute kidney injury. I'll explain this term in detail in a few minutes. And the question is, should you go ahead and give antibiotics and contrast studies and so on? What about the kidney, despite the fact that the creatinine is 1? Is it bothersome? In the second case, we have the usual patient coming into the emergency room who essentially wants to go home, but they have pneumonia. And it's possible that they would be given antibiotics and discharged home, but the lab test comes back as an abnormal creatinine. And this may refer to the fact that the patient is volume depleted because they haven't been eating particularly well, or it could refer to the fact that the patient is in the initial stages of sepsis and kidney damage as a result of sepsis. Perhaps pneumonia resulted in sepsis that caused kidney damage, acute kidney injury. What would you do? The patient wants to go home. Neither of these cases really stretch us intellectually. They are managerial problems, because in both cases, really what you need to do is wait 24 hours to make a decision. And 24 hours is not plausible with the current rules about emergency rooms. We used to keep patients even for days to make decisions, and that's not legal anymore. So the problem here is that a single measurement of creatinine does not give adequate diagnostic information. Let's take a few examples of clinical cases to see why it's not as articulate as it should be. Here's a newborn case. This is our work in the neonatal intensive care unit. This is urine measurements in a baby. And you can notice that the creatinine is rising in the baby, typical of kidney damage, but you can see that the scale is in days. And it really takes a number of days for the creatinine to rise, especially to a level that it comes to clinical concern. And the reason is that without a placenta, creatinine normally rises in a newborn. But here it seems to go up and up and up. So in this particular case, it was day 6 or 7 that the child came to the attention of the neonatologist who was concerned about kidney injury. In this case, we have a child undergoing surgery, and you can see it's many days later that the creatinine rises to threshold. This is 3 days later. The surgery is over here. And so here we have a problem that creatinine has to accumulate in the blood. So all kidney diagnoses are always made in retrospect. You can't make a diagnosis in one measurement straight off. It has to be serial measurements seen over time. The second problem here is that it's a relatively insensitive test. This is an early case of obstruction of one kidney. You can see the kidney has a little bit of edema around it. The ureter is beginning to be enlarged here. And the problem with this kidney is that certainly this is a damaged kidney, yet the creatinine doesn't change very much from normal patients to that damaged kidney existence. And there's a reason for this, because the other kidney can hyperfilter. So creatinine is an insensitive test. And in fact, it's said you need more than 50% of loss of renal mass to get a creatinine elevated. This is a study from kidney donors between 20 and 37 years out. And these individuals only have one functioning kidney, yet their creatinine is up around 1.2, which is the upper limit of normal, as a mean creatinine. So creatinine is very insensitive to the loss of even one kidney. You have to reduce the other side further to get a bump in creatinine. And lastly, creatinine is not necessarily the most specific marker, and the reason will become clear in 2 minutes when I show you some clinical data, but suffice it to say these are two patient populations, and I'll tell you who the patients are in a few minutes. And in one case, we have 1,391 genes that are activated, in the other case, 84 genes that are activated. And I have to tell you that both of these populations have exactly the same creatinine. So on a genetic level, there are reasons to elevate. There are different seemingly genetic categories, but yet they both increase the creatinine to the same extent. So when your patient comes in and you find a rise in creatinine, we all consider this tantamount to organ failure, that the kidney's no longer functioning as well as it should be. And so a rise in creatinine is really downstream of a very lengthy sequence. And you can see that when it comes to our friends in cardiology, they know what they're doing decades before than in nephrology. Look here, organ failure in heart disease after a heart attack is defined as wall motion abnormalities, that the ejection fraction has decreased dramatically in the heart. And this is called organ failure. But surely we don't diagnose myocardial infarctions this way anymore. We actually, in my day as a resident, we used to look for EKG changes, which were more sensitive than wall motion abnormalities. And even here, this is a very insensitive test because there's a lot of MIs that are not transmural. And so starting 20 years ago, there was the beginning of a movement to find a biomarker in the blood that was released as part of the myosin-actin complex that was released in reflection of ischemia to the heart. So you can imagine if you look at this time course here, or severity course, called the severity, that patients could be troponin positive, EKG negative, echo negative, yet we still say they have an MI. This is incredibly important because we have to triage patients immediately. Other patients who are more severely affected will be troponin positive, EKG positive, echo negative. And the worst situation, most severely affected patients are troponin positive, EKG positive, and wall motion abnormalities as well, representing organ failure. When it comes to creatinine in the kidney, it's our only way to measure kidney function and kidney dynamics. And so we only have an assay at this stage. And as I told you, it's very insensitive and delayed. Couldn't there be markers upstream? And these would be the so-called biomarkers upstream of this. Now, having said that, you have to realize there's something very important to realize when it comes to biomarkers. And I just told you the example in troponin. Couldn't it be that you could have a cell damage marker that's positive, but the creatinine is not elevated? Because creatinine is insensitive and delayed, right? And I don't know that you've gone down this full pathway to organ failure. Just like you can have troponin positive, but EKG negative and echo negative. I can tell you that this flow diagram is incredibly important, and the literature is filled with a lot of mistakes, not having considered that damage and biomarkers represent stages of a pathway. So if we agree that there are stages of a pathway leading to the most severe diagnosis, organ failure, we also have to agree that these sequences may be different for different stimuli. This is a stimulus called volume depletion or prerenal azotemia, and it's corrected by saline. Is it the case that cell damage organ damage. These various steps are identical from just volume depletion and sepsis. Are these really the same entities? Or are we going to get different biomarkers? So my field is a very confused field out in the literature because very few people have just sat down and said, what are the obvious steps? And using the traditions from cardiology as a model. Some of these ideas have been recently put into this fourfold table. This is a very well-known table in nephrology. And what it says is that there are two things that one can lose with disease. One can lose the function of the organ, but it's not really damaged. That's what we call volume depletion, because in volume depletion, the laboratory tests, the current ones, look like the kidney's damaged. But it's not really damaged. It's simply responding in a physiologic way. Loss of function means that it doesn't remove as much creatinine as it did before because it's a volume-retaining organ attempting to resuscitate that patient. So this is a new category of disease based on the fact that you have functional markers, so-called functional markers, or I call end-stage markers like creatinine, and you have damage markers. So you can see that this is a fourfold table based on two ideas. Creatinine, whether it's elevated, it's the filtration marker, the function marker, or proteins coming from the damaged tubule, and you can have four possibilities. So this is what we used to call volume depletion. Damage without loss of function means taking out one kidney, donating a kidney, because creatinine may not rise. And lastly, if creatinine is elevated and damage markers are present, then you have the worst condition. We used to call this acute tubular necrosis. This lands people in hospitals and on dialysis machines, this last compartment over here. So there's a little bit of sophistication beginning to occur in our field, dividing things into this fourfold table. I like the kinetic image better than the static image, but it has a lot of the similar ideas encoded in it. Okay, if you were going to make biomarkers and do something with them, what would you need to do to establish a biomarker? And for any diseases that you work in, you would have your list of possibilities, I'm sure. And here are my 6 criteria. Be aware that in nephrology, very few people have spent the time throughout all 6, except that the protein I happened on has a lot of biological interests, and I'll tell you about that in a minute. That would be number 6 on the list. But you can see, obviously, you'd want to do these things, these 6 things. So we are a lab that's interested in kidney growth, and I purified a protein that was a red protein, and it's called NGAL, NGAL. It's a lipocalin. It's a beta-barrel protein. It's a soluble protein. It's in the blood and it's in the urine on damage. And I was told that it was in the urine on damage by a friend who happened to come upon it on an Affymetrix gene array. And I was studying it, and I gave a lecture on the subject at Albert Einstein. And the gentleman said, look, this is the top protein expressed in kidney damage. We had it as some type of growth factor, a long story. I said, okay, let's take a look in our patients in the intensive care unit. So here are a group of patients here. This is each lane is a urine sample. This is our standardization curve. And you can see some patients have a lot of this protein and some patients have a little bit of this protein. And we always know this protein because we cut these bands out and sequence them. We are very thorough in each one of these things. And we know it's right above the standardization curve because this is non-glycosylated NGAL protein and this is glycosylated human NGAL protein. We find exactly the same measurements when we compare this to a commercial tool that had been invented with our collaboration. There are some cases where there are multiple bands on these gels, and so we've felt it's important over the years always to do Western blots. So we've Western blotted maybe 5,000 patients so far. Around the world, the NGAL test has been applied to probably 30,000 patients by now through automated means, ELISAs and so on, and kits and machines like this, the Abbott Architect. We stick with Western blots because I have to be sure I'm looking at this protein, which I'm calling the monomer. These are dimeric forms of the protein or heterodimers. and I'm not sure what their value is to the epidemiology of renal disease as opposed to the basic protein at the monomer level. Okay, so what about this protein? How does it compare to renal damage? When do we see it? You can see in the blue curve, this is the NGAL protein that rises extremely quickly on injury, and in the black curve, the creatinine. So temporally, it beats out creatinine by about 2 days in these neonates. It's a very wonderful window not to have to wait. In fact, on the first day of our measurement, which was day 2 here, it's already quite elevated. This paper we put into Lancet. It's from Prasad Devarajan in Cincinnati Children's, and these are kids undergoing cardiac surgery. This is when the creatinine reaches threshold on day 3. Here's the NGAL coming up at 3 hours. So we have a temporal relationship with the inciting stimulus that's much better than creatinine. It comes really, really quickly, 2 to 3 hours. Okay, having seen that, what I decided to do was to get 4 medical students together here. And I sent them to our emergency room and to the emergency room at Staten Island General, the emergency room at the Allen Pavilion, and in collaboration to an emergency room in Berlin, the Charité Hospital, in collaboration with Kai Schmidt-Ott, a colleague and friend of mine. And what we did is we collected 1,600 something samples of urine. We simply collected urine. Take 1 drop of urine, we shook the patient's hand, we said goodbye, and we were going to analyze their clinical history and compare it to the NGAL measurements at the point of contact in the emergency room. So here are the results of this paper. The data has been published in Annals of Internal Medicine and a journal called JACS, Journal of American Cardiology, College of Cardiology. These are a scale of creatinine. I just told you that creatinine is insensitive and delayed, but the problem in our field is we have to compare things to creatinine since that's the only marker of kidney damage we have, even though it's a thing we wish to replace. So a lot of these comparisons you'll see in the literature and we'll start to think about are really quite circular in reasoning, but this is what we need to do. So this is a patient who has had a 1.5-fold increase in creatinine, a 2-fold increase in creatinine, and a 3-fold increase in creatinine. And that increase in creatinine could occur from the emergency room to any day up to day 7 in the hospital. And so in this drop of urine, if the patient will have a rise in creatinine up to 1.5-fold their baseline, we get this level of NGAL. 2-fold their baseline, we get this level of NGAL. 3-fold their baseline, we get this level of NGAL. And we get a beautiful correlation as a dose-response curve. Okay? The other biomarkers that are out there have dose responses, but interestingly, they seem to flatten as the disease gets worse, and we think we know why, and I'll explain that a little bit later. So we have a very beautiful logarithmic dose-response curve. This is reflected in the AUC-RC, which is the comparison of the NGAL number to the exact creatinine that the patient will obtain. You can see that if you only look at patients with rifle F, if you segregate these data and compare the NGAL versus the creatinine, you get an AUC-RC of 0.83. If you look at all comers, including the low increase in creatinine, you only get an AUC-RC of 0.72. Here's NGAL in the top curve compared to the other biomarkers. What does this mean, this increase in AUC-RC? What would that tell you? And here's what I think. Remember this pathway. So in mild disease, rifle R, for instance, small change in creatinine, you can still have a lot of cell damage. It's just that you haven't damaged enough of the kidney to raise the creatinine very much. So the biomarker is positive. The creatinine is only weakly positive or negative. If, on the other hand, the damage is very severe and you go all the way to organ failure, then the NGAL will be positive and the creatinine will be positive. And so the worse the disease, the better the AUC-RC. The less worse the disease, the biomarker will be separate. And so it's extremely important to understand disease severity, to have a dose-responsive biomarker and compare it to a standard like creatinine. To understand, you may not go down this whole pathway. Here's some of the data from these large studies. So these are patients who have kidney disease that's going to be unremitting and potentially need dialysis. We call this acute kidney injury. Here are patients with just, they're dehydrated and need volume. This is the difference in NGAL levels. Here's the creatinine. The creatinine's elevated in all groups because it elevates in all these groups. Chronic kidney disease, it elevates. Volume depletion, it elevates. Acute kidney injury that's damaging the kidney, it elevates. And this is the spread of NGAL. Here's a dose response with the timing of NGAL. The longer NGAL, the creatinine's elevated, sustained, the higher the NGAL level. And this is 1 drop of urine in the emergency room. Okay. We repeated the study just to be sure of the data. Here are patients with liver failure. Their kidney function is depressed in all of these categories. And here's the NGAL. It rises in acute kidney injury, damage, but the volume depleted group, much less so. Even though creatinine is elevated, this is the inverse of creatinine. It's the same thing. Even though creatinine is elevated in all cases. Creatinine is elevated in volume depletion, chronic kidney disease, hepatorenal syndrome, and acute kidney injury. But it's only the acute kidney injury that has the NGAL. The data has been confirmed by another group. The numbers are shown at the bottom. We did it again. We asked the question, what about this volume depleted group? Does it really have less NGAL compared to the kidney damage group, the intrinsic damage group? These folks will need dialysis. These folks need saline. That's all. Saline in the emergency room and go home. And these guys may need dialysis. So there's a big difference in NGAL levels. Okay, so we have a lot of clinical data that says that the protein comes really quickly. The protein is dose responsive with injury. The protein is specific to tubular damage of the kidney. I'll explain that in a minute. And it's not present if you just simply are missing a few liters of fluid from your body. A lot of people wanted to know more about where this thing came from, so I decided to make a luminescent mouse. What we did is we stuffed a luciferase into the ATG of the NGAL gene, and then we infarcted the kidneys. And so this is a living mouse expressing NGAL. You can see the NGAL because the gene has been replaced by luciferase. And so if you damage the ES cells, the embryonic stem cells that we made the mouse from, you can see that when you damage it, you get the luciferase, no damage control. You can see that if you take cells from the kidney and add bacteria to them, you get a huge increase in the NGAL signal. If you treat with antibiotics, you can suppress that. These are living mice, so you can follow them over time. And they have a beautiful NGAL signal with ischemia. That's creatinine following later. It's dose responsive. The more ischemia you give, the higher the luminescence. Here it is in the urine. And if the kidney's really, really damaged, you get more RNA for NGAL than you do for actin. This is volume depletion. NGAL is negative in volume depletion in patients. It's negative in the animal. There's no NGAL in the kidneys. In the animal, that's volume. You can see the animal losing weight. You can see it getting smaller and smaller in the animal. And lab tests showing it's volume depleted. So we can measure it. We can understand its temporal relationship with creatinine. It comes before creatinine. We can see it's very dose dependent on the damage. We can see it's specific to real damage, not to the saline responsive volume depletion, the simple thing that you and I go through every day. And so let's look at some of the biology of it, because if that checks out, that helps us think of this as a biomarker. So we did a mouse, we did ischemia, we looked at in situs, and the blue is NGAL protein, RNA that is. And what you can see is that it has a very beautiful patterning of NGAL expression. Some of these parts of the kidney are the thick ascending limb, and parts of these kidneys, the salt and pepper kind of patterning here, are these cells, and these are called intercalated cells. They are a small percentage of the cells of what's called a collecting duct. This patterning can be seen with markers of these intercalated cells like the proton ATPase, but no one would have thought that these cells would turn positive when you have ischemia of the kidney. You see much of the attention of biology of ischemia of the kidneys focus on these areas that don't stain for NGAL, which are the proximal tubules. But here we're showing the thick limb of Henle and these intercalated cells, express a protein on ischemia. Turns out every time you damage tubules, you can get NGAL. This is in the disease of HIV of the kidney, where you get NGAL expression in these microcysts in the tubules. So NGAL comes from kidney tubules, and it comes from parts of the kidney that were not suspected to be activated in ischemia, which really encouraged us to really do some more experiments. Here we decided to infarct half of the kidney, and that's where NGAL is expressed. The other half of the kidney was normal and was not infarcted. Okay, so we have a protein that comes very early before creatinine, and it comes from a strange part of the kidney, an unexpected part of the kidney. Okay, how do you prove it definitively comes from that part of the kidney? Maybe it comes from other cell types in the body. How do you definitively prove it these days? And the answer is that if you think something comes from a cell, you should knock it out. Knock out the cell, I mean. So it turns out that the other part of my lab, Max Worth and N-Dun-Q, had discovered in my lab, and Kai Schmidt had discovered in my lab a way of doing this, of actually finding genes that we can knock out and we can throw away cell types from the kidney. Let me just briefly say two things. So this is early kidney mesenchyme. These are stem cells of the kidney. We found a method to convert them into nephrons. In finding a method to convert them into nephrons, we then did a gene array every day. When we did a gene array every day, we found all the transcripts, all the RNA that was activated in this conversion process. We then looked at all those genes and found 78 transcription factors. We took 78 transcription factors and put them into adenovirus. We put the adenovirus back into the stem cells. One of those adenoviruses containing one transcription factor caused the conversion. The transcription factor is a new transcription factor called CP2L1. It causes nephrons to form. And that's a very lengthy, obviously, discovery process. But when we knocked it out, the intercalated cells disappeared. These are intercalated cells marked by a number of their functional proteins, like the ATPase, carbonic anhydrase. When we did the knockout of this transcription factor, they're gone. The blue cells, in this case, the red cells, in this case, are completely gone. And if NGAL comes from those cells, then NGAL should be gone in the urine. And this is the result. So when you knock these cells out, you lose NGAL. So that proves, this was published in JCI, so that proves that these cells are an important source of NGAL. Maybe not the only source, but an important source. So there's a microscopic relationship with damage. When we infarct the kidney, we see NGAL in the infarcted area. And when we focus and see special cells, when we knock them out, NGAL's missing. It's a huge amount of effort to prove something, but no biomarker, every biomarker really should go through this process, in my opinion. There's a lot of stuff out there that I'm not sure of. So why does the kidney make so much of this protein so quickly on damage? What's the real biology here? And what about these funny intercalated cells? So how are they involved? And why, if you calculate the amount of NGAL in the urine, it's actually gram loads of NGAL are made in a damaged kidney. So the question is, well, what's it doing there? Why do you need so much? What does it do? And here's part of the answer. This is NGAL protein, and it has a beta barrel. And the beta barrel has positively charged amino acids in it. And those positively charged amino acids bind to this structure. And this structure is recognizable because these are catechol rings, dihydroxybenzene. But what was not recognizable, at least by me, by other people, was this backbone. Roland Strong, the crystallographer, when he expressed this by recombinant means, got a red protein. We did too. I called him up one day. I said, why is the thing red? He said, because it has iron in it. But it's not iron bound to the protein. It's iron bound to an organic molecule, this one, embedded in the protein. And that organic molecule got in there when we cloned the protein in bacteria. This is a bacterial substance. This is called enterochelin. This is the way that bacteria eat iron from our body. Bacteria make enterochelin. to provide these hexadentate oxygens to chelate iron at an affinity of 10 to the minus 49 molar. And it binds to these metals, obviously mostly to iron. And then the bacteria recover this and break these bonds with an esterase. And that's how bacteria rip iron from any protein in your body. This is much higher affinity than transferrin, ferritin, or anything else. And so how does NGAL get in there? Well, when a bacteria enters the system, it stimulates the production of NGAL. And then NGAL is secreted and provides a positively charged calyx that exactly binds these catechol rings and takes this molecule away from bacteria, starving the bacteria for iron. Here's the protein when it's just loaded with iron, with its so-called siderophore, bacterial siderophore, and here's the protein without it. And this is what happens if you add NGAL to growing bacteria. They stop growing. It's an iron-starving mechanism. Is this unique in biology? Not exactly. That's the reason we think we have circulating transferrin and ferritin, and the regulatory hormone hepcidin is probably all about sequestering iron from bacteria. So here's a specific example, and this differs than other examples because this binding is so specific to enterochelin, which is the siderophore gram-negative organisms. It's a very specific pathway. So this is a molecule that's critical in the biology of tissue damage, it turns out, because it is an antimicrobial that we think protects the tissue from infection during damage. The fact that this molecule is critical, and in fact, when we knocked it out, urinary tract infections hang out longer, is a type of molecule you want as a biomarker. Think of the pregnancy test when you measure beta-HCG. That's an essential molecule in pregnancy. A biomarker of the kidney should be an essential molecule in the damage sequence. And this is an essential member of innate immunity, is what we came up with. The FDA has approved this molecule for use in animals to test nephrotoxins by various companies. This is a letter of intent. It's on sale in Europe. I hope one day it will be on sale in the US. I can talk for great length about the patenting, the legal issues, the response of one's colleagues, writing letters, and all kinds of crazy stuff when it comes to getting out there with, this is the first kidney biomarker. There's another group that says, well, they have a better one. We don't really agree. But I can talk for length about that at some other time, perhaps. Finally, the FDA has recognized our work, at least in animals. It should be in humans, too, and I'm hoping that will happen. A competitive biomarker is on sale now in Europe and in America. It's a combination of biomarkers that we are having trouble with. So this is the NGAL protein. These are clinical samples. And you can see NGAL is positive in one patient and negative in the other patients. But this new biomarker is positive in all the patients, including normals. So we're a little bit confused. This is a very, very intense area, and with tremendous competition and false backstabbing, false reports, and I'd be glad to talk to you about that in length when I'm finished. So there's many unanswered questions. We make a lot of NGAL to protect ourselves against bacteria, gram-negative bacteria, but suppose there are no gram-negative bacteria there. And ischemia is a sterile disease. Maybe there's an endogenous ligand. How is NGAL expressed so quickly? 2 to 3 hours? It's not the fastest gene in the world, but it's called probably a delayed primary response gene. It's pretty fast, 2 to 3 hours. You know, what's unique about this, and what about these cells? And each of these topics I'll just spend 1 minute on addressing, because I think it's central to the idea as to whether this is a biomarker and what's really happening here in the background. There is another ligand. We discovered that our urine is filled with these small catechol molecules. That's catechol, not enterochelin, which is 3 of these held to a backbone, but the individual units, the functional unit of the enterochelin, which is called catechol. They can fit in the pocket of NGAL. This is real crystallography. This is not a model. And they fit between these positively charged amino acids. 3 of them will find there. The reason you don't see the other 2 is they fell off during the preparation of the crystal. We know they're there because we changed the color to exactly what enterocheline looks like. That has 3. This has 3. This has probably 2 inside the pocket, and colorless would be 1. We found these molecules by simply doing a little chromatography assay in our lab, which you all did in high school. You spot something here, you run water in this direction, and look, iron doesn't go anywhere unless you add your favorite substance, and we find catechol binds to iron and moves it, solubilizes iron. There's enterocheline. It's a real winner. There's catechol and other compounds. And we screen urine and so on, and we purify from urine, and we find these small molecules present there. So there's probably an iron scavenging role of NGAL beyond enterocheline, even in the absence of enterocheline, I mean. Why is it expressed so quickly? So there may be an endogenous iron scavenging function beyond the presence of bacteria. Why is it expressed so quickly? This is a little complex, but I'll just spend 1 minute on it. Here's our gene. Here's our promoter. Here is the insulator that stops transcription. This is an NF-kappa B site. Bacteria bind to toll receptors, turn on NF-kappa B. NF-kappa B binds to the promoter, and away we go. I'm not going to show you all the data. Maybe we'll just come to the model here. It turns out that if you take the NGAL locus, even without stimulation, guess what? Polymerase II is already at the start site. A variety of histone modifications are already present. This is an open chromatin configuration. And when NF-kappa B binds, it doesn't have to do a lot of the preliminary work that many genes require for transcription. They're already there. So what NF-kappa B does is it adds a novel histone mark that is involved in the movement, releasing polymerase II to move down the gene. This is called reversal of pol II pausing. It's a mechanism that's been recognized in a number of genes. And what it says is that the gene is ready to go. That's how ready to go. Ready because it's already loaded, and the to go is a switch with NF-kappa B striking this. There's other steps here, but for simplicity purpose, it's ready. We just need a signal, and within 20 minutes, 30 minutes, we're moving. By 3 hours, all the machinery is complete, and we're pumping out NGAL messenger RNA. So it's expressed so quickly, like I told you, for the biomarker purpose, because it's not that it's coming from somewhere else. You see, if something comes in 2 to 3 hours, your colleagues will say, this isn't real. It's coming from the blood, or it's coming from a cell damage. No. It's actually the architecture of the gene allows it to move really quickly. Is NGAL unique? What about the cells? What about the uniqueness? What are we going to think of next? Okay. Is NGAL unique? Here we go. So my colleagues around the world have met in various committees. They believe that any time a creatinine is elevated, it should be called acute kidney injury. It's called the KDIGO guidelines. The KDIGO guidelines reverse 50 years of renal physiology because they do not accept the concept that simply a change in creatinine could be due to toxic or non-toxic events. This is incredibly important to me because NGAL only comes during toxic events, not during non-toxic events. So we decided to take 2 sets of mice, and I'll show you how this is going to finish here. We can take 2 sets of mice. And 1 set of mice, we're going to clamp the renal arteries for 10 minutes. That's real ischemia, real damage. In the other case, we're going to take food and water away for 72 hours. In both cases, the creatinine rises from 0.2 to 0.3, 0.38. When we look at the tissue, there's no damage in the volume-depleted mice. Just like you and me, we've had an illness, a diarrheal illness, a fever. We go to the emergency room, our creatinine's elevated. By the way, the physician should give you fluids and discharge you, not admit you for dialysis. This patient here, these mice should be admitted for dialysis. Look at the kidney. There's an infarcted zone right here. We are confused because they both change creatinine. And according to KDIGO guidelines and all kinds of national things, that's all you need. So we decided to take these mice and look at the genes that are expressed. The ischemic genes are expressed in the middle of the kidney. The volume-depleted genes are expressed in the pelvis of the kidney. There's a different distribution in where the genes are expressed. So they can't be the same disease. The genes are different. They can't be the same disease. Now, this is an interesting test. I'll show you a few of the genes themselves and how they shake out. But the technology we use is called laser capture. We just cut out sections of the kidney and just did RNA sequence and measured all the genes. We can do better than this, but this is a good piece of evidence. Okay, we looked at the genes we got in ischemia. They are genes of the fibrin clotting cascade, the TLR signaling, the JAK-STAT, and so on. It's all the kinds of genes you would expect in a damaged tissue. Look at the volume-depleted genes. None of them were positive. None of these pathways were positive. Only the positive pathways was gluconeogenesis, the TCA cycle, amino acid transport, as if the kidney were starving. But that's exactly what volume depletion is. We took the food and the water away from the mouse and got exactly metabolic genes back. Whereas the ischemic genes were the typical cell death, cell damage, and cell repair genes. They're not the same. None of the signal transductions are the same. Either your standard, the hippo pathway, the Wnt pathway. Okay, here are some examples of genes. These are genes that we've selected because they are not secret genes. They've all been published in the literature at one point or another. These are genes that turn on. Look at this, 49-fold increase, 214-fold increase with NGAL, clusterin 32-fold, KIM1, another gene, 500-fold increases, absolutely no increase at all in volume depletion of all of these genes. In fact, we have 1,000 genes that increase in one state. And then we asked an interesting question. There are no biomarkers. No one has considered volume depletion to have biomarkers. Why would they not consider that? The reason they wouldn't consider that is it's only considered a form fruste of ischemic disease. But remember, KDIGO lumps everything together, so this is just a weak form of AKI according to the current thought. This is the real thing. This is a form fruste, but they have different genes. So we said, well, maybe they're biomarkers of volume depletion that are independent of ischemia. And we came up with 10 genes that turn on in volume depletion. They do not turn on in ischemia. And in fact, when you thirst the animals there, the gene goes up. When you give hydration back to the mouse, they come right back down again. There's obviously volume depletion genes. They're metabolic genes. They're the things I mentioned. Those are different than ischemia. Here are some of the new genes. So here's present in ischemia. It's not present in volume depletion. Same creatinines in these guys. Right? Some of this is human samples now. In ischemia, not in volume depletion. Ischemia, not in volume depletion. Here's a new gene that comes along the collecting ducts there of the kidney. Here's a gene that's present in prerenal, but not in normal or not in ischemic damage. It's the opposite. No one's thought of this because no one considered this to be anything different than the ischemic disease. What about this lousy technique of laser capture where you pull out genes from a swath of tissue? Is that adequate for modern day biology? Well, in some ways it's a good start because you're collecting regions. It sort of gives you anatomical regions. But there's something more interesting that we can do. So we've invented a mouse that allows you to label RNA in any cell you want, specific cells you want. So if you'd like to label the intercalated cell, we have a way to put a tag on those RNAs and just to pull those right out of the kidney. So we only look at intercalated cell RNA or principal cell RNA or proximal tubule, whatever you'd like. And then we say, well, okay, if we can pull out specific RNAs, what are the responses to prerenal azotemia versus ischemia? And you can see they're very different genes. When we pull out from the principal cells really respond to volume depletion. Prerenal azotemia is called. They don't respond with many genes to ischemia. Look at the intercalated cell. It's exactly the opposite. These two cells sit right next to each other in the collecting duct. They're having opposite responses to two different stimuli that were previously lumped together. I've decided that one thing I'd like to do with my money is to make a map of the kidney and kidney damage. That means inventing various Cree drivers for different parts of the nephron. We've done that. And applying this. Is this expensive work? It kind of is, because we need to do RNA sequencing, but I rely so heavily on Paolo Guarneri of the C2B2, and a wonderful assistant professor, his name is Nicholas Tatonetti, of bioinformatics, who has helped me tremendously put this work together. And so we can march up and down the nephron and say, what changes in a disease? Are these diseases the same? What changes when you give a medication? Or does the entire nephron respond, all the cells respond equally? Or is it they're targeted to one area? So lastly, I'd like to point out that all of the work I presented to you came about by mistake. I happened to purify a red protein. I happened to give a lecture. Someone raised their hand and said, this is the highest thing in ischemia of the kidney. It happened to be in intercalated cells, kind of specifically. The wrong place for ischemia, but it led us to say, well, we need to map the whole kidney. So you can see a progression of ideas there based on the fact of semi-disbelief that it shouldn't be this way, but so let's map it out and find it. These intercalated cells turn out to be more interesting than I would have known. I tell you something, intercalated cells were essentially discovered and characterized here at Columbia University by a nephrologist named Kais Alicati, who was the previous chairman of the division. And these are cells that pump acid into the urine. One of the best therapies for a UTI is to acidify the urine. And these are the cells that pump acid. They also make NGAL. And in looking in the literature, guess what? They also make interleukin 18, which is a biomarker, in fact, of kidney damage. It's coming from the same cells, it turns out. Interleukin 18 also calls forth neutrophils. It's a chemo-attractant for neutrophils. This RNA-7. RNA-7 is a powerful antimicrobial against fungal species and a number of bacteria. They're coming from intercalated cells. It turns out that we found a cell that's typical of host defense. These are important cells to stop UTIs. Probably UTIs and evolution were the things that would most commonly drive kidney damage. And here you have a whole response system sitting in the bottom of the kidney in the collecting ducts, ready to go. The mapping of the kidney is just time and money. And it seems to me we're seeing that cells do different things in different conditions. It's important to understand the kidney. The other reason this is important, as I summarize here, is that if a patient has an elevation, let's just go back to the mice as sort of an absurd group of patients. But if patients have an elevation of creatinine, this would make rifle R criteria, by the way. This both make rifle R criteria. They have acute kidney injury according to the current standard. Let me tell you what I would get on my cell phone in that case. I would get a call to dialyze these patients, both of them. But this patient just has volume depletion, just needs saline. There's no kidney damage. So mapping the kidney and really understanding the damage markers versus so-called functional markers is essential to refining our clinical thought process. I can tell you the debate over this has gone up to the chairman of medicine. Patients come in. We say no to dialysis because it's pre-renal. The intensivists, the cardiologists say we must do it. They go behind our back. They call our chairman. They bring us in. And we start dialyzing patients with volume depletion just to get rid of the political problem. We need to understand these things like we used to. You give saline for volume depletion, and you worry about dialysis for intrinsic damage. Okay, so let me summarize. Biomarkers are incredibly important because they change our definition of acute kidney injury. We can't lump things together when a biomarker is positive or negative. We have to make decisions. NGAL has non-creatinine characteristics, in fact. It's immediately expressed because POL2 is bound to the promoter. It's very sensitive. You don't need a bump in creatinine to see it because that's end-stage kidney. That's damage to more than 50% of the kidney. We don't need that. We can see it. It's very dose-responsive. It responds to tissue injury very well. These characteristics refer to the promoter. It's hardwired in the gene. The saline responsive form of elevation of creatinine is not the same as the saline non-responsive form. It's coming from non-traditional locations in the kidney. And AKI mapping, this mapping expedition with RNA sequencing in different diseases and different medical therapies, I think is warranted. Biomarkers will not only move our field ahead, but they'll change the way we do medicine in the following way. A motorcyclist comes to your hospital and has a creatinine of 1, and you want to do some fancy tests which could damage the kidney. The creatinine is 1, but an astute clinician would say, look, this creatinine of 1 is just delayed. It's going to go up because the kidney is damaged from muscle damage. What would you do? Well, if the biomarker is negative for acute kidney injury, then there's going to be low risk. If this creatinine of 1 represents acute kidney injury, NGAL will be very high at that point. What about the patient that you want to send home or wants to go home with some fluids, but you're worried that this is actually septic damage to the tubules? This is not volume depletion, septic damage to the tubules. And in fact, the creatinine is 1.5, which is clearly abnormal, but you don't know which one it is. What would you do? Well, if you had a volume depletion pre-renal biomarker, if it's positive, please give the patient salt water and discharge them from the hospital. However, if that one's negative, but NGAL is positive, which is your tubule damage marker, then please, no salt water, and admit the patient for potential dialysis. We finally have a way to see specificity by knowing genes coming from each and every cell of the kidney. Is this personalized medicine? I think it actually defines what could be personalized medicine, because each type of damage is going to have a different signature on different cells of the kidney, different RNAs. And I think that this is actually, this seen in mouse models, humans is going to change, and it's going to make very specific diagnosis where now we just look at the creatinine and just say, admit the patient for dialysis. So my lab is an old lab. I've worked with a lot of great people. I have a small lab. It's usually 4 or 5 people. The data I've showed you is probably the output, 1/3 of the output of my lab in the past 5 years or so. And so I thank you for having me here.